I'm Steve Hanlin, the entomologist in charge of controlled pollination here at the station. Because of the process we use in production of seeds growing them in a caged environment, we must have insects to pollinate the flowers. Honeybees in a small nucleus or nuc hive fill that gap in seed production. In a beehive, there are three castes or types of bees: a worker, a queen, and a drone. The majority of the hive is made up of worker bees. The worker takes 24 days to develop from egg to adult bees. Its function starts out with cleaning and caring for the brood and other bees, the queen, and the drones. Later in its life, they protect the hive and care for the larvae. The final stages of the worker's life is to go out and collect nectar and pollen from the plants. In the summertime, this lifespan is approximately about a month and a half from hatching or emerging to death. In the winter, a worker will live up to several months before perishing. The next, and possibly most important bee in the hive, is the queen. She develops from egg to adult in 16 days. Generally, there is only one queen in the hive. Her sole purpose is to lay eggs for the hive. She also puts out a smell or pheromone which designates that hive specifically to the worker bees. The final caste of bee in the hive is the drone. A drone takes 32 days to develop. Its sole function in the hive is to mate with the queen. There are approximately only 200 of these male bees compared to 20,000 worker bees in a hive. The other two, the worker and queen, are both female. We use a 6-frame hive or a small hive for our cage pollination. This makes it much easier to put this smaller hive into cages for pollination rather than the larger 10-frame standard hive colonies of bees. Each of the nucleus hives contains approximately 2,000 to 3,000 worker bees and a laying queen. The queen grafting process is actually a 3-day process. We must first find the queen of the colony and contain her. This is done either by placing her in a queen cage or the frame she is on into a separate box, being careful to make sure that she stays with the colony she comes out of and does not get put into the nucleus hive. The method that we use for our queen production is called the Doolittle method. It consists of manipulating frames of drawn comb to attract young nurse bees who care for the larvae and the queen cells. Drawn comb is a sheet of foundation which has the cell stamped on it and gives the bees a pattern to continue to draw out. It is used for the storage of honey or pollen or the production of brood. They build on the structure and produce the rest of the wax on the frames so that the cell can be used for honey storage and for the larval bee production. To prevent the bees from going back to their original colony, which many of the field bees will do, we must take this nucleus hive to a new location where it will remain for 1 to 4 days queenless, before having a queen cell introduced into it. The process of producing queens, also known as grafting, is much the same as what a colony of honeybees will do in the case of the death of the queen or a replacement of a queen that is less productive or is injured. We use worker larvae to produce our queen larvae. In the first 3 days after hatching from an egg, there is no difference between a worker larva and a queen larva. They are both fed what is called royal jelly. This is a special substance that is produced in young adult bees and is fed to the young larvae to start their initial development. After those 3 days, the difference between the worker and the queen arises. A worker larva is fed bee bread, which is a mix of pollen and honey, while the queen continues to be fed royal jelly. This difference in feeding also determines the development time of each of these bees. A queen takes 16 days to develop from an egg to an adult. A worker takes 24 days to develop from egg to adult. Also, the worker develops in a horizontal form, and the queen develops vertically. On the first day prior to our grafting, we do a shifting of frames so that open young brood or eggs are put above a queen excluder and drawn comb beneath the queen excluder. A queen excluder is a metal barrier that is placed between supers of hives which traps the queen so that she does not get into our newly grafted cells. If she does, she will destroy them. Above the queen excluder, we have placed young open brood or eggs. Below the queen excluder, we have placed older larvae and sealed brood. Also in the top box, we have placed our frames of queen cells, which we will be grafting. This is to allow the bees to polish or to clean the cells up before we place larvae in them. If the cells are dirty, the bees will remove the debris. They will also remove the larvae and will not allow those queens to develop. At this time, we also go to a separate hive and look for a specific frame that contains larvae that are 24 to 48 hours old. This is because they are still being fed royal jelly, and these are the larvae that will best be accepted by the bees for queen production. The following day, we take the frames of 24 to 48 hour worker larvae and place a single larva into a polished queen cell. The larvae are C-shaped, and in removing them from the worker cells, they must be lifted gently out of the cell and placed into the queen cell in the same way. This is because the larvae have spiracles or breathing tubes on top, and if they're flipped or bumped and damaged, the larvae will then suffocate and are promptly removed by the worker bees, and those larvae will not develop into queens. On the same day, we make up what we call the swarm box. This is a box which we place the young nurse bees, which we have drawn above the queen excluder. It has no openings, so the bees are trapped and they're forced to begin pulling out the queen cells over a 24-hour period. Once the frames are grafted, they are placed into the swarm box for 24 hours. The box is placed into a dark room and the bees are allowed to start caring for the queen cells. Once the queen cells have been started, they will get better acceptance and continue to be drawn out after being placed back into the cell builder. The next day after grafting, the frames of queen cells are taken and placed back into the cell builder. This allows the cells to be surrounded by the young brood and eggs, which also means that they are surrounded by the nurse bees. This setup guarantees that most of the bees will be young and will continue to care and feed royal jelly to the cells and draw them out. The cells will remain in the cell builder for 10 days. After 10 days, the mature queen cells are removed from the cell builder, and that frame is replaced with an empty drawn comb. The cells are then removed from the grafting bar and placed into the queenless nucleus hives. The nucleus hives with the queen cells are left alone and not disturbed for 14 days. Once the queen emerges, she is considered to be a virgin queen, unmated. For approximately a week, the queen will move about the hive, taking small flights, possibly 6 feet away from the hive, and then coming back. After about 1 week, the queen will go out on 1 to 2 mating flights. This is a flight out to a designated area where the drones and queens congregate for mating purposes. At this time, the queen will mate in flight with approximately 20 drone brood. Upon completing her mating, she will return to the hive and in most cases never leave the hive ever again. After 14 days, they will be checked to assure the queen is mated and is laying eggs. Once this is determined, the nuke is ready to be placed into the cage for pollination.